after describing well-fed state and starvation state in terms of what the hormones will be released and what will happen to each of the tissue types that control metabolism, let's actually delve into starvation just a little bit more. Now, I give you two different charts here, or two different tables from two different books, but the energies are similar, and in terms of the probably percentages, if you actually calculate it, they're almost identical. But what we are doing is we're going to compare and contrast where the energy sources are available if we don't intake any food at all. And so at the top of the list, we look energy in terms of calories. That's interesting because that's an English terminology, although we talk about calories in terms of our eating here in the US, mass in kilograms. And so this is a 70 kilogram man that has typically well nourished, and so these are average data. In the bottom, what I wanted to keep was the idea of kcals and then kilojoules, so you could see that. But what we're looking at is the largest source, which you already know, is the fats. It is mainly in the adipose tissue, and the calorie count is very large. Now, here's where the issue comes in when you start looking at this. It says that if you calculate a basic amount of calories that you use in a day, you do a simple conversion, we would typically have 83 days of energy if we had this many calories in 15 kilograms at a 1700 calorie per day intake. Now, the metabolism doesn't allow us to extract energy for that long. The second largest amount of energy is protein. Now, that what we're talking about is skeletal muscle breaking down, and so as you break down skeletal muscle, then you can't or you lose the ability to move around when you get to the end of starvation, and you can extract lots of energy from that. Glycogen is the store that we use both in skeletal muscle and in liver. And so way back when, I told you that skeletal muscle had the highest amount, okay, but in terms of mass per calorie, the most concentrated amount is in liver. Skeletal muscle, again, does not give up fat, does not give up sugar, and so this 600 calories will be used in the muscle, and that can extract 8.4 hours of work based upon a very simple calculation. Glycogen liver is not intended to be used by glycogen, but to be released to feed the blood to make sure that it is at a constant concentration, and we have about half the amount of energy as we do in all of the muscle. The circulatory system is, in itself, has mobilized energy sources in all different forms: glucose, fatty acids, and triacylglycerols, the fats themselves. So this is free fats being mobilized. This is fats that have not been integrated into the adipose tissue yet. And we can grab that energy and use that. And so we have almost 100 days, although under starvation conditions, there's no way that we could reach 100 days of not taking in any food. What happens in terms of the body processes when you undergo starvation? Before you even get to starvation, what you tend to do is, even in normal conditions, is after extended times of exercise, the body will start to mobilize fats and mobilize protein degradation. And that's for the muscle and exercising. If you aren't taking in any fuel, any foods, especially carbohydrates, then the operators will tend to shift both of these working. The reason is this: we're going to send fats down into acetyl-CoA, but acetyl-CoA will not be able to enter the citric acid cycle very well unless there's oxaloacetate. Now, the signal to send fats and to degrade proteins, and this is in the liver cell, tends to also enforce that the oxaloacetate is going to start up gluconeogenesis. And so we will be sending out glucose to make sure that the brain is functioning. Okay, so if fats are coming in and acetyl-CoA is trying to enter the citric acid cycle, then if there's no oxaloacetate to do that, this builds up in concentration. Two things occur. You can take the amino acids and you can send them into oxaloacetate to both synthesize glucose as needed, but also to fund the citric acid cycle. And so some of the proteins are used in that particular nature. Now, this is still inhibited because of the limitation of the starting material. So what happens is acetyl-CoA normally reacts very easily with oxaloacetate to enter in the citric acid cycle, builds up in concentration. And so we start sending ketone bodies into the bloodstream as a very high energy molecule in the bloodstream. And so we're sending glucose for the brain exclusively. We're sending ketone bodies, which can be used by our other tissue types, including muscle. We're breaking down fats, okay, but we're also creating urea at this moment in time. Now, based upon energy consumptions, there's a whole number of ranges that you can see. Most dietary comparison charts, all the things that are on the labels of boxes for your food are based upon a 2,000 calorie a day intake. But depending upon what you are, you can have very low consumption or very high consumption of calories depending upon what you're doing. And so if you look at just the numerical value, we have somewhere between 1 and 3 months worth of food or energy molecules, but they're in the wrong forms. Here's the important factor: blood glucose levels cannot go below 2.2 millimolar. Normally it's 4.4 millimolar, and a lot of times hospitals won't use these particular concentrations. They'll use milligrams per deciliter or 100 mils to measure that. Our first priority, again, to keep glucose going to the brain. One other thing I'd like to talk about, just as an aside, what happens when you undergo the Atkins diet? Atkins diet is a way to lose weight very, very rapidly. And what happens is you intake lots and lots of protein, and you intake lots of fat, or you can intake fat as well. What you do is you limit the carbohydrates. So what you do is you give your body calories, but you give them in ways that the body doesn't quite recognize. And what happens is the lack of carbohydrates coming in makes the body think it's under starvation. So it will start sending out glucose into the bloodstream to keep the brain going. What will happen is, since proteins are coming in excess, there is no way to store them. So if you are going to degrade proteins and sending them in, then you have to go through the citric acid cycle. And to fund the citric acid cycle, if you are sending glucose this direction and not providing glucose for acetyl-CoA, then what you're going to do is you're going to mobilize fat stores, and you break down all the fat that you eat along with your proteins, and then you are mobilizing fat from your adipose tissue, and so you can lose quite a bit of weight. But what happens is you're setting yourself up for failure in that your metabolism is set for a starvation condition, even though you're bringing in calories. As you can lose lots of weight, and you can maintain that weight for a long time as long as you keep the diet. As soon as you go back to a diet that includes carbohydrates at any significant amount, the body has a memory, it has a trigger, And so once it sees glucose, it'll start utilizing it in the ways that it intended it to be, and it'll start storing glucose. It'll store glucose in the form of glycogen, which it hadn't had very much of. And then what it'll do is it will actually convert the glucose into acetyl-CoA, not sending it to the citric acid cycle, but sending it to fat synthesis. What happens is your body has felt like it's been under starvation conditions, and so it is going to try to protect itself in the future by adding more fat, because it anticipates you're going to encounter another starvation condition. And so your basal weight, or your set weight, actually goes up. Unfortunately, many people, not just the Atkins diet, but many people diet rapidly with very odd types of diets, most of them very restrictive, and so the body undergoes what it feels or interprets as starvation conditions during the diet, and then goes back to a normal diet. And what happens is people lose weight and then gain it back and gain a little bit more, which then makes them want to lose weight. They'll lose it again, and then they'll gain a little bit more. And so your set weight continues to rise and rise and rise. If you look at starvation, what happens over the days, 0 days, what you tend to have is very high concentrations of relatively of glucose. There is fatty acids mobilized, and this fatty acid concentration is pretty stable. And then your ketone bodies, there is ketone bodies at all times, but not significantly. Now, if you take a look, this is 0 days, and that is 10 days. Here's 5 days. Okay, by 5 days, you are going to have fats mobilized at almost the highest level it will be. There is a minimum level of glucose concentration that your body will be sending out. And look at what's on the rise, ketone bodies. Ketone bodies will become the largest source of energy in the bloodstream after, and it crosses over at about the 10th day. And so after that, ketone bodies and basically the fat stores, that largest amount of energy will be floating around and being utilized from ketone bodies. And so we don't increase the fat, because what we're doing is we're breaking them down within the liver at about the fastest rate that we can. And so actually sending more fatty acids into the bloodstream doesn't actually increase the rate of synthesis of ketone bodies. All right. Now, when you use your fuel, each fuel type that you have has a different rate of consumption and also a total amount. And so when you take a look at what can happen when muscle contracts, the actual concentration of ATP in the muscle is used first, followed by phosphocretin, we talked, followed by muscle to lactate. That's a fastest conversion, but again, incomplete conversion, a very small fraction of the energy. Okay. Once you're there, lactate is a dead end product. Muscle, if you take muscle glycogen, the sugar that is there, and give it to CO2 in an aerobic type of reaction, which is slower, the rate now is less than half of that of the lactate. All right, you can exercise and you can extract a lot more energy. It's the most efficient in this particular pathway for glycogen. Now, what happens if you are using all of the sugar in your cells? It'll start to extract it from the bloodstream. The liver will then mobilize glycogen into glucose into the bloodstream, which then is transported to the tissues. It has to be transported in and go down the entire pathway to break it down to make ATP. Rate even slower. What is interesting, though, is that the glucose coming from the liver and the adipose tissue coming from the fat have about the same rate, but there's a ton more energy. And so, how long does it take to use these resources here? 3 seconds. 3 seconds of intense workout uses up all your ATP. Phosphocretin gives you a whole another 6 seconds. The glycogen and an anaerobic, 3 minutes. Liver glycogen, 50 minutes. Now we're talking about real amounts of time. Muscle glycogen, that is aerobic, hour and 25 minutes, and adipose tissue over 5 hours. If you are going to run a Olympic timed sprint, you're doing it in closer to this time period. Okay, we have broken the record under 10 seconds, but you can see that at that moment in time, you're using the fastest sources, the amount of conversion of the energy at its fastest rate to convert. So 73 plus this is immediate, and after that, you cannot run as fast or at that rate for very long periods of time because you have to use other energy sources. And so by the time an Olympic runner is running the marathon, what happens is you transition from uses of the sugars like from glycogen stores into about a 50-50 mix of protein and adipose tissue while you are actually doing or finishing up the race. Now, what people have done to increase the rate at which you can run for very long distance runs, there's often, if you see on long runs, people handing out water. That's very important. You're losing water and you want to make sure your electrolyte concentrations are balanced and you're replacing the water that you're sweating out. But now you're also seeing gel-like pastes given to the runner. These are glucose pastes or sugar pastes. This allows them to have another source of sugars, of carbohydrates that can be readily accessed, that isn't being depleted from the liver. And what happens is the liver will lower it down and keep the concentrations lower so it can extend it. But if you are adding these supplements in terms of extra carbohydrates during the race, you can extract energy faster in that respect than you relying on the adipose tissue in and of itself. The next item that we're going to focus on is how we signal. Now, we've talked about hormonal signals, but what we really want to do is remember that almost all of our hormonal signals are a result of a neurological signal from the brain, whether it be, and it's mostly on an unconscious level that does these things. And so let's talk about how the nerve impulse is going to cause us to have small molecules released and to control all of these events. Now, the neuroendocrine system includes neuronal responses, which are very, very short action, short distance, cell to cell, and hormonal, which is classified as long distances. And that usually includes the release of the small molecule in the bloodstream, which then actually goes over the entire body, but may only affect one tissue type because the actual tissue that this hormone is being released has the receptors for those, and the other cells do not. Neuronal and hormonal are similar in action. Molecule released, a response will occur, but timings are different in how fast they can process. Hormonal control is working through receptors, whether it is on the surface of the cell, which we talk about signal transduction, and you get the benefits of not only the signal, but also amplification, adaptation, things of that nature. Usually relatively very quick responses in the body. The other is through the receptor becoming a transcription factor upon binding of the response molecule. This particular type of response is usually much slower, and so a drug of that nature may take hours to days, where a receptor on the surface could be as little as, in an asthma attack, you take the inhaler, the albuterol binds very quickly, and within relatively seconds, you can have a response. The types of hormones that are released can be classified in several different ways. These are three classic ways. The endocrine, it's released into the bloodstream, goes over the whole body, and can interact in the whole body. The pancrean is a release into an extracellular space to affect something neighboring, so very close, but not through the bloodstream. And the autocrine is a hormonal release that comes back and affects itself. Sorry. Now, hormone classes. What are the types of molecules? Well, we've been talking about insulin and glucagon. Somatostatin is also in the mix, but you may not realize they are actually peptides. And so usually from 3 amino acids to 200, relatively small peptides are released in the bloodstream and act as a hormone class. A lot of times one peptide equals one hormone, but there are several classes in which the peptide will be broken down and become several hormones. Insulin is a very good example of one that we know very well. You have the pro-insulin, the signal sequencing to make sure that that protein is placed into the correct vesicle before it is going to be released. Now, proinsulin is a fully intact protein after the signal sequence has been cleaved. There are numerous disulfide bonds. The full activation is a cleavage to get a mature insulin, which is 2 chains now, which were once one chain, but that are linked through disulfide bonds. In early treatment of diabetes, the source of the insulin was from pig, and what they did was they harvested the tissues, they extracted the insulin, and that was the source of the insulin. And now using molecular techniques, we can grow insulin in cell culture, isolate them, and you can now buy human versions, but the human version is from a cell culture type of development, and you can also still find porcine that is used, and even down to the traditional isolation methods are still used, but the genetic molecular biology processes are widely used. Hormones can also be small organic molecules or even large organic molecules, and there are 2 classifications. The catecholamine, which are water-soluble compounds that include one that we showed at the beginning, epinephrine, and the eicosanoids are fat-soluble. They were ether lipid defined, and remember the eicosanoids produce prostaglandins, thromboxanes, and leukotrienes, all from the arachidonic acid. That's a 20 carbon fat with 4 unsaturations. If you take a look at epinephrine and go back one, you get norepinephrine, and then before that is dopamine. Dopamine is a neurotransmitter, and if you go back, dopa, or you will hear L-dopa, is a drug that is given quite often. And then one step back, you are now at tyrosine. And tyrosine can be converted from phenylalanine. So these natural hormones are derived from an amino acid conversion. Looking at more hormone classes, we looked at the steroid based hormones, very hydrophobic, sex driven hormones, derivatives of cholesterol, usually bind receptors in the nucleus. Vitamin D, again a fat based hormone, derivatives regulate calcium. Vitamin D, a derivative of cholesterol, and we need to have the excitation of sunlight to cleave the ring system, and then if you remember, 2 hydroxylations from 2 different tissue types before it becomes fully activated. The retinoids, derived from beta carotene, regulate cell growth, survival, and differentiation. thyroid compounds. The thyroid compounds are relatively interesting, and they control energy yielding metabolism. So they control the metabolism and interact with the metabolism that we've been looking at in the past few chapters. Thyroid hormones are small molecules that are iodinated. And if you look at that, you don't think of too many foods that have iodine. In actuality, the government has regulated that any table salt, if you actually read the bottle, it's iodinated. It is regulated to have iodine placed in it so that we have a source of iodine in our diet. Thyroid hormones have iodine in part of the organic structure. And so this is kind of an oddball case. One interesting fact about your thyroid and iodine: when we were first dealing with nuclear or atomic weapons, the government would issue a anti-radiation pill. Well, what the anti-radiation pill was basically iodine salts. One of the worst byproducts of nuclear fallout is iodine-based radioactive material, which is a gamma emitter. And if you're exposed to any sort of radiation event, the fact that iodine gets in your system is just one of the many different types of radioactive material that you could absorb. What is really bad about it is that it's a gamma emitter, which is high in energy, and the fact that iodine, once in your body, is concentrated into the thyroid. It's a very small organ. And so what they did was they gave the iodine pills, usually potassium iodine, and you would take it, and what you would do is you would put a high concentration of quote-unquote clean iodine, and any radioactive iodine that you encounter, even if you absorb it, will compete out and not be absorbed in the thyroid. Now, what unfortunately was thought of is, at that moment in time, is, oh, I've got a pill, it protects me from radiation. Not all radiation, just one very specific type. There's one signaling molecule that's in the body, nitric oxide or nitrogen monoxide. It is a gas, which is, we've never seen a gas as a stimulant before, but it diffuses through the membranes. It's nonpolar. It can work through short distances, and the radical NO stimulates guanine cyclase. And so we had a lot of receptors that bound the G-type receptors that will upregulate adenylate cyclase to make cyclic AMP as a secondary messenger. These will upregulate GMP, cyclic GMP receptors, and cause the same types of interactions. Now, we did see a cyclic GMP pathway in rhodopsin in our eyesight, and so these types of pathways are seen as often, sometimes as cyclic AMP. I'm sorry. Okay, so we're going to send out these signals. What we'll do is we'll pick up here and we'll talk about how we actually regulate those tissue types that produce the hormones, and then how those will go off and signal other tissue types to excrete other hormones as we go through a hierarchy of control.